Okay, thank you. Some really interesting talks. So we're going to start a panel discussion. If I could invite all the speakers to the chairs table. Thank you.
Okay, can we have first question? Behind you.
Ask my question first. Oh, thank you. I had lots of questions because those were really, really interesting talks. If I can find a page in my book that I wrote them. So the first one is for Mujgan. I'm going to ask all of them, and then you can ask them. Okay, Mujgan, you said that you didn't see any resistance development in the bacteria towards your phages. I wonder, did you do something where you checked to see whether there was a recurrence in those plants that you had treated? So you said that you infected the leaves, you put the phage, and then you measured the bacterial load. Did you go back to see whether there was a resurfacing of infection in those plants? And if you did, and you treated them again with the phage, did you see that there was less susceptibility the second time round? That's a question for you. The second question was for Celinda. So Celinda, you said that I really liked the cool stratification story that you gave. I thought that was really brilliant. You said that the patients who didn't respond to maybe I'll say it all wrong, but you know what I mean, who didn't respond to the aspergillosis, LPS or something like that, were the ones who later on went ahead to have aspergillosis and potentially died from it. My question is, did we check residual aspergillus carriage in these patients before they came into hospital? That's one question. Second question is, it's looking like if you go into hospital with some of these conditions, you might end up with aspergillosis. Can we use that stratification to find out who these patients might already be before they go into hospital, so they don't go into hospital and catch nasty infections, and maybe we can treat them at home, and then maybe they won't get aspergillosis? Sorry, let me see. I think I have one more question. I was paying attention. So, this one is for the person online. Adora, you said you saw an increase in Acinetobacter baumannii in the very young population, and then in the ICU patients that were that were older. I wonder, did you see any other infections in these patients, and was there any correlation in the fact that if they got that infection, they would have Acinetobacter, or was it just Acinetobacter that you found?
Right, I guess I could start. If I remember the question. So yes, so in that slide, I very briefly mentioned about the field work, but that was a year of work that went to that in one slide. So in that field work, yes, so we infected the trees with the Pseudomonas stringi, and at each time point, so we infected these with phage after. And at each time point that we collected samples, we re-isolated the population of the pathogen that we put in. And then we tested those pathogens for the sign of resistance by sequencing, and also by testing the phages against, the wild-type phages against the bacteria that we re-isolated from the field. And so during sequencing, we didn't see any mutations happening compared to the reference genome. We also found that the wild-type phages could kill the bacterial population completely, so it means that the resistance didn't occur. But in the treatments that we had phages on, we couldn't see any pathogens at the end of the season, so this pathogen population completely died off. So we've done a thorough test to understand if the pathogen resistance. So we did like different methods of testing those against. We didn't go back to that field in the next year because of the funding and because of the end, but our plan is to do another field work hopefully soon and to do all the tests and be going with more questions this time to see if, again, resistant images or not.
See if I can remember mine now. So in terms of whether the patients just had Aspergillus, so in Cardiff they have a just constant test approach. So, you know, they're constantly doing PCR. Basically, if there's any sign of fungal infection on sort of the first sign of suspicion that they have it, then they treat. So basically, there were no signs before that there was Aspergillus. In terms of can we treat patients before they go to hospital or know, you know, that they have a high chance, I think absolutely, yeah, we could test them before they come into hospital. I don't think we would be able to stop them from coming to hospital because these are hematology patients, they have to come in for treatment, but they could be put on prophylactic therapy straight away if we knew before they come in. Yeah.
We had a question for the speaker online.
Okay, thank you. So, yes, why the infection was more prevalent in the young and adult ICU was because in such areas, there's usually increased use of invasive devices like the nasogastric tube for feeding in this set of patients and urethral catheter, because most of them are non-ambulant, and also some of them too may have had other invasive procedures such as ventricular shunts. And yes, the patients had other infections. However, most of them came in with other comorbid infections such as being managed for hypertension, patients that had stroke, from hypertension. In the process, they contracted Acinetobacter baumannii infection. Others were being managed for diabetes mellitus, and there were some that had surgery. Like I said earlier, some had surgeries, like those that also came in due to trauma, road traffic accident that needed a shunt or intervention. Thank you, Ma. I hope that answers your question, Ma.
You spoke about two kinds of resistance genes. One gene that would take care of one antibiotic and one gene that can take care, that can counter multiple antibiotics or antifungals, sorry. How common are they, and are they found in other members of the Candida species?
So that was a kind of a theoretical framework to think about it. These are not genes, is this related to the cross-resistance?
Yes.
Yeah. So it's a theoretical way of thinking about how cross-resistance works, that there is a single mechanism, I don't know if it's a single gene, but a single mechanism that'll enable the organism to survive both drugs. We don't know what those are. It could be something as easy as efflux pumps, right? And that's definitely the first thing we're going to test. But otherwise, we don't know what the mechanisms could be. Yeah.
Questions for Mochkin. The first one is, how do you consider that the bacteriophages might affect the bee themselves, or do they not? And the second one, a little bit weirder, being from here where there's lots of rain, how does wet weather affect how the bees carry the bacteriophage? Like, does the wet weather eradicate it, or does it survive?
Yes, so we are at the moment doing a lot of tests to understand if phages impact the bees itself, and for example, bees' foraging behavior, bees' microbiome, and so on. So we know that phages are very host specific, so they only infect stringy, because we tested the phages against a range of microbes that we know that they exist on bees, and also like a beneficial microbes. So we know that phages, they don't impact those. But we're also going to do some microbiome study on bees itself to see if the application of phage impact those and behavior. So this is ongoing extent. So next year we're also going to do some field work with the phages to answer the second question, to see how wet weather or like rain impact the bees' movement. But considering that cherry trees are usually planted in polytunnels, so they are usually covered, so we know that the wet weather shouldn't impact that much in that term. So, but we're going to test anyway to see how different like environment factors going to impact the delivery of phages by bees.
So, hello. So my question is also for Adele. So I have very distant but yet fond memories of whenever I was in your lab working on antifungal tolerance. And I was wondering if you could tell us a bit more about it. I think I remember like, at the time it wasn't genetic. We were thinking it might have been like epigenetic. And I was wondering, could you tell us a bit more about how do we get around something that we can't really sort of like pinpoint down to like a gene, and like how the unique sort of problems that arise compared to true resistance?
Yeah, this is the million-dollar question. You know, it's really, really difficult to understand what's happening in tolerant cells, and it really comes down to the single cell. So we're trying to do some single cell analysis now to understand, because in a population, there's a small percentage of cells that will undergo this, and there are strains out there where that percentage will change. So it could be 90% of the cells might be tolerant, whereas in other strains of the same species, it could be 5%. And so we're trying to dissect out those. So we did transcriptomic analysis on tolerant cells, the high tolerant versus low tolerant strain. And what we see is it's basically a shutdown mechanism. So most of the translation machinery are down. And the ability of the high tolerance strain to get into that kind of quiescent state is better than they believe the low tolerance strain. So it really is akin to the persisters in bacteria. We've also identified some transcription factors that seem to be kind of the master regulators of this based on the RNA-seq data, and we're looking to do more experiments now to see what do those transcription factors, you know, are they really essential for this process? In terms of epigenetics, I've always, you know, I've come from this from an epigenetic point of view, because if it's not genetic, it must be epigenetic, right? And definitely when we disrupt epigenetic genes, it does influence tolerance. Yeah, and that's what your project was, right? If I remember. So we do know that there's an epigenetic component, but yeah, getting down exactly to is that direct or indirect, I don't know. Thanks.
Can I ask another question?
I have a question for Thomas. Sorry. Was there a question somewhere else as well? Oh, go ahead.
Hi, staying on the... Oh, don't know, working. Staying with the crop fungal issue, how much impact is that having on human fungal, and what's the transmission?
Yeah, so for Aspergillus, it's really well worked out, because Aspergillus, we're all breeding in Aspergillus spores right now. So, and Aspergillus is common everywhere. So there is a very well-defined pathway from the crops, from the agricultural setting to the clinic in terms of Aspergillus spores. They can travel a wide variety. They gain resistance on the farm, and we'll all just inhale them. So that's kind of quite clear, where the epidemiologist studies are trying to figure out exactly how much of clinical resistance we can attribute to the antifungals in the crops, but it's really a focus of the fungal community right now. In terms of Candida, it's less clear. For Candida transmission, you need to physically touch them, right? So you need to be exposed, you need to be, be on a farm or be close to someone who's been on a farm. However, we've done studies now, and we're only, just showing this. We can take fruit from Tesco, and there are Candida on there that are resistant, right? So they pass through the food chain, and we are being exposed. How much of that is actually showing up then in the clinic, we don't know. But Candida, there is environmental reservoirs of Candida. There are environmental reservoirs of Candida that are resistant. And yeah, so in theory, it can affect us in a clinical position, but yeah, we're only just looking now, so I'll keep you posted.
More questions?
Yeah.
Right, so I have a question for Thomas. So it's very interesting to know that you can disrupt or kill bacterial biofilms with plasma. but I don't know, what is the size of the system that you have which generates plasma? And in that context, what do you think about the applications? Whereabout you can use, for instance, can you have a gun which simply shoots some plasma and kills bacteria in, I don't know, some area of interest here?
Thank you for the question. So we have devices which we call plasma pens. They are about the size of a pen, but some of the bigger ones would be, I guess, he does a lot of wet lab work, about the size of a 96 well plate. And we also have very small ones about the size of the palm of your hand as well. These are used medically, and so they're designed to use it. People have used it for crazy things like, it's a big area in tumor, so people use it for like brain tumors, and they use it for prostate cancer, which is the obvious choice. We do a lot of work on skin and bone infections, so we would use it surgically as a tool as well. And our big emphasis has been like, we don't think plasma is going to replace antibiotics, but it could definitely be a very good supplement as well. So that's how we use it.
Hi, I've got a question for Mojigan. Did you test the time period for which the phage was viable on the field? And the second question, actually I have two questions, sorry. Did you test, as you mentioned you would study the bees' behavior, but did you notice any reluctance with the bees when applying the phage, and how long did it take to overcome that?
So for the first question, yes, we had different treatments in the field experiment. So one was the phage on its own, and one phage with the host that is a pathogen. So when the phage was on its own, there was no host infection first. So phages couldn't survive after 3 days, so we couldn't detect anymore. So it means that, but when we infected the plants with the pathogen first and then applied phages could be detected up to 30 days. So it means that phages could only survive if the host is present. But because it's obvious because the phages use the host to replicate, basically. And as I mentioned in my talk, that we try not to use a very high dose of phage to kill the bacterial population completely, or eradicate it, but just to reduce the bacterial population. So it means that we still have the bacterial population for phage to use and replicate. So that's what we try to do. But also in terms of when we talk to farmers, one of the questions is what happens to the phage when the pathogen is not there anymore? So we know that the phage cannot survive without this host. And then for the second question, we haven't seen any impact on the bees' behavior so far. So there's no reluctance in phage going to the infected plants, or delivering phage, or moving phage in between. So we've seen that bees are quite happy as long as they see the flowers. They are happy just going straight towards those. So in that sense, we haven't seen any foraging impact on the behavior, foraging behavior, also on visiting what flowers and which flowers. But next experiments that we're going to do, hopefully in the polyton, is going to tell us more about that aspect as well.
Do we have time for one more? No, we do not have time for one more question, sorry. So, can I have a round of applause, please, for all the talks and panels? And thank you to our online speaker as well. Hopefully you can hear us.
Thank you.